Good morning, everybody. First of all, I'd like to thank the organizers for giving us this opportunity to present this exciting work that we've been doing with proteogenomics. For those of you who have been following the work on Galaxy Galaxy for Proteomics research, we have been working on using transcriptomic as well as genomic data to look at some of the novel proteoforms that have been expressed by various organisms, and I'll be presenting that work. First of all, I'd like to thank, or at least acknowledge, quite a few of our collaborators. We collaborate with quite a few researchers from University of Minnesota, and there are quite some of them that have been mentioned here. Professor Matt Andrews from University of Minnesota-Duluth. We work with him, and I'll be presenting some of his work in this talk. And Dr. Laurie Parker, with whom we have started looking into some of the data-independent analysis data that we've been looking into. I'd also like to acknowledge Professor Tim Griffin, who's the PI on this grant and who has been extremely encouraging and the reason behind most of the proteogenomics as well as metaproteomics work that we have been doing using the Galaxy platform. We work very closely with the Minnesota Supercomputing Institute and the Galaxy P team, including James Johnson, who is here, who has actually helped us a lot, not only in maintaining the Galaxy infrastructure, but also developing newer tools and putting some of the concepts that we have in mind in place as workflows. We also have collaborators from Johns Hopkins Institute, Karen Reddy Lab, and Mo Hiderian is here. We have been working with them on one project, and I'll be talking a little bit about that. We also collaborate with a lot of researchers, developers across the world. Beyond Groening is here. We work with him for quite a lot of things, including the Docker installation and Docker work that we do. We have been funded by NSF and NIH. With that, I'll get into the talk, and I'll be basically using these four bullet points as a way to convey this work that we've been doing for proteogenomics. I'll briefly mention about proteomics. I will talk about what are proteoforms, bring in the concept of proteogenomics, how we are using transcriptomic as well as genomic data to identify some novel proteoforms, and how Galaxy has enabled us to look at some exciting results that we have been getting. I'll take you through some of the example proteogenomics projects, and then maybe we'll also have a look at what we are looking in terms of future. Where is the field right now in terms of Galaxy research in proteogenomics, and where can we go from here? So for those who have not been initiated into proteomics, I'll give a very brief crash course in proteomics. Proteomics, as you know, is research that we perform to identify expression of proteins, as well as detection of these proteins in tissue samples or in any study that you are performing. We start with proteins, end up with digesting them into peptides, and then separate these peptides using chromatography methods, ionize them using the mass spectrometer, which is our main instrument that generates the data. Mass spectrometer ionizes these peptides, fragments them, and then you get what are called as mass spectra. These mass spectra, if you look at the second part of or the bottom part of the slide, these mass spectra are matched against standard protein databases. So these are protein FASTA files that one matches them against using search algorithms, and then you end up identifying these spectra as peptides. Now, if you match these spectra against a reference or a database of known proteins, you end up identifying known proteins. However, in last few years, researchers have also started using not only standard databases, but also RNA-seq data that is generated from the same sample that you're analyzing for proteomic studies. The RNA-seq data, in this case, is converted into protein FASTA file, and then you can end up identifying proteins which are specific to your sample. And this also includes some of the novel proteoforms that could be present uniquely in your sample. As you are aware, in cancer or any other physiological studies, there are quite a few genomic rearrangements that take place, and these genomic rearrangements and their expressions are then captured by this proteomic data. So there was a term that was introduced in 2013 by Neil Kelleher and his lab. They basically came out with a term called as proteoform. This was to bring in the field of proteomics and genomics, or at least explain the origin of protein with respect to a gene-centric approach. And what a proteoform is it describes a molecular form in which a protein product of a single gene can be found, and this encapsulates all the genetic variations, splice isoforms, as well as post-translation modifications. And so when we're using RNA-seq data, and that's what I'll be talking a little bit about today, we would be looking at novel proteoforms, or proteoforms that have not been identified before because they're not present in the standard proteomic database. So in 2014, we came out with a basic structure of what we thought was a good workflow or sub-workflows that we could use for proteomic database search. And this includes generating databases. This includes generating spectra that can be used for database search. So in the second module, you see spectra that are generated from your instrument are matched against data, let's say from your RNA-seq data that's converted into protein FASTA file. When you match these spectra against your peptides, these peptide identifications are parsed, and then you can subject them to BLAST-P analysis. And the idea for using BLAST-P analysis is to identify anything that has not been reported yet. Once we have them, we perform what is called as peptide spectral match evaluation. So we go and look at the quality of the peptide spectral match and ensure that what we are reporting is indeed of good quality, and then eventually put these peptides onto the genome. Now, all of these have been published in some way or the other. For example, for database generation, we have modules that have that can take in RNA-seq data and generate a protein FASTA file. This was published in 2014. And then we also have various database search algorithms that are present, including some commercial ones and some open source ones. And eventually, after all of this data processing is done, we visualize all of these peptides on a genome to ensure that the genomic variation that we have been observing. So central to all of these workflows are two main tools that we use. One of them is BLAST-P workflow. What it basically does is takes your putative novel proteoforms or peptides, subjects them to BLAST-P search against the NCBI database of the organism of your choice, and then eventually you end up identifying novel peptides. So anything that does not match to the NCBI, latest version of NCBI database, you would report that as a novel peptide. And then once we have them, we end up doing a peptide spectral match. So we use a tool called as multi-omics visualization platform. There's a tool called as MZ2 SQLite that was constructed with the help of James Johnson as well as Tom McGowan at Minnesota Supercomputing Institute. What it does is it generates a SQLite database from the database search that you have performed using MZ-identimal files in your workflow, and it captures all the information about the peptides that have been identified. One can use that information, filter out the interesting peptides of your choice, in this case, this will be novel proteoforms that one is looking at, and then eventually look at only those peptides that you are interested in. So you could look at the peptide spectral match and its quality, visualize them, and then ensure that these are indeed of good quality, and only report those instead of reporting anything that you think is of not good quality. So with these workflows, I'm going to talk a little bit about some of the example projects that we have been working on. We initially started with a large data which we had published, wherein standard proteins were identified using SALIVARY proteomics. This was a database or a data that was generated using 6 individuals, which were pulled together, and so SALIVARY supernated from these 6 individuals were collected. It was subjected to a lot of fractionation, so various methods of fractionation were used in order to ensure that you identified low abundance proteins. And then these were acquired on an instrument called as a thermo LTQ Orbitrap instrument. And there were as many as 200 raw files that were generated, which is a large amount of data when we're looking at since this was published in 2009. Obviously, the standards have changed quite a lot in the last 7 years. We searched this database against a 3-frame cDNA database, and after all the filtering and all of the workflows that we used, we reported as many as 55 novel peptides. Now, that might not seem a lot, but given the amount of stringency or the abundance of caution that we're using, we were reporting only those that we were very sure of, because one, we were doing it for the first time, and secondly, we wanted to be sure that what we are reporting was something that was reproducible. So all of these rearrangements could be classified into various categories that have been shown here. So out of these 55, we interestingly found that 14 of these actually fall in a region called as PRB1, PRB2 region. And this is a region on chromosome 12. It belongs to these proline-rich basal protein regions. So these are highly abundant proteins that are present in saliva. It turns out that these proteins have some kind of a role in adhering to microorganisms and then clearing them up. At the same time, they might have some protective mechanism in attaching to toxins that are introduced into the oral cavity. We do not know the reason why we're finding so many frame shift mutations in this region, but there could be a role wherein for these proteins in maybe encountering some other microorganisms or any other protective mechanism. So this is still under investigation. The other interesting study using workflows or some modification of these workflows was using, so there's a researcher named Matt Andrews, Professor Matt Andrews from University of Minnesota-Duluth. He works on ground squirrel, and his study is on looking at hibernation proteomics of ground squirrel. So as you can see, what they had done was they had collected samples of animals that were from in fall as well as during hibernation season and those after hibernation season. The graph here basically shows the blue line shows the temperature of the room temperature at which these animals were kept, and the black line shows how the temperature changes, fluctuates during the hibernation as well as pre-hibernation season. So samples were collected, and the samples that were collected were heart muscle and skeletal muscle samples. And interestingly, and these both have been published, and I encourage you to look at them, to look at some of the details that have been published. But in heart muscle, we ended up identifying as many as 162 novel peptides or novel proteoforms, and again 162 across all the 3 replicates. There were a lot more identified if you take all of them together. One of the reasons for this has been that the genome of this organism, though sequenced, has not been assembled yet. So it's still present as context. So there are quite a few errors in terms of annotation assembly that resulted in identifying so many new proteoforms. In the skeletal muscle, which was another published study that came out this year, there were quite a few interesting set of proteins that were identified. What I maybe forgot to mention was this data also had RNA-seq data associated with it. So they used both RNA-seq data as well as proteomic data to make these conclusions. One of the conclusions that they made was when you looked at the proteins that are expressed in the active phase after the ground squirrel came out of hibernation, there were proteins related to glucose metabolism that increased sharply and fatty acid metabolism that decreased. So that was one of the observations. There were also some observations in relation to the muscle proteins, myosin protein, and fast twitch as well as slow twitch isoforms, and how that changed depending upon the phase of hibernation. The other study that we have been working with is with Karen Reddy's lab, and this has been a study, again, has been published. There was RNA-seq data available for this. There is proteomic data available for this. The study that was published was for correlation of RNA-seq data with proteomic data, but Dr. Reddy's lab, along with Mo Getiria, wanted to follow up some of this study using long non-coding RNA sequences. So long non-coding RNA sequences are known to be sequences which are more than 200 base pairs in length, and they apparently comprise as many as 80% of the RNA that's expressed in a tissue or an organism, but they're known not to be coding for any genes. They seem to have properties for mRNA function, but they do not have properties to be an open reading frame. So there were classes of long non-coding RNA genes that were generated, and then we did a 3-frame translation on that, and using our workflow, we looked at how many of these were identified. And we could find a list of quite a few of them identified here. Obviously, this needs to be followed up. So this is more at the discovery stage. Once the lab starts looking into the details of this, we are hoping that there's some interesting biology that comes out of this. The last study that I'm going to mention is a study that we have been doing with Dr. Laurie Parker's lab from University of Minnesota. And this study is, again, has got RNA-seq data as well as data in form of what is called as swath data. So swath data is data which allows you to perform quantitative analysis across the sample, and that's a new method that has been kind of emerging in proteomics, and we're kind of excited to correlate both the quantitative data coming from swath or data independent analysis and RNA-seq data. What they had was they used the cancer cell line, and which was sensitive to drugs, and there are these 3 drugs which are kinase inhibitors that they had used. And what they did was they passaged these cells in increasing concentrations for almost 3 months until they came with cell lines which are resistant to these drugs. At the same time, they did a control experiment wherein they kept passaging these cells, but in absence of any inhibitor, so they also had a sensitive cell line. They collected RNA-seq as well as proteomic data from this, swath data from this, and they have already done some studies in terms of correlating the RNA expression to protein expression, but they are also interested in looking at some novel proteoforms that are getting expressed. And so this is basically something coming from, this is a chromatographic peak of one of the novel proteoforms of peptides that was identified. And this particular peptide is actually quite close to a RAS oncogene family of proteins, which seems to have effect on cancer phenotype. And what you see here are basically expression levels in control, which is K, and I and D are for the drug levels. So if you normalize these samples, this is what it looks like. One on the left side is the bar chart of what a sample should look like in normal case, or when all of the samples are used together, while you can see that the expression of these novel proteoform is decreased in the control and is in variable amounts, but in an elevated amounts in drug-resistant cell lines. So there is some interest in following this particular work and finding out what this exactly means, and there are quite a few other novel proteoforms present as well. With this, I come to my last slide, but I just wanted to mention here that now we have come to a point in proteogenomics wherein the interest is not only in identifying novel proteoforms, there's obviously an interest in that to find out what these are and what it means to the biology of the organism, but there's also an interest to make a correlation between mRNA expression as well as protein expression and see how that correlates and how that addresses some of the biological questions. Keeping that in mind, we have actually started working on both database generation, genome, proteome, quant integration, and working on various tools to ensure that we could, one, enable that, and secondly, also visualize these results that come out of the analysis. So this is a busy slide, but there are quite a few new developments that we foresee from these interactions that we'll be having with various projects. Lastly, I would like to acknowledge the researchers who made this possible, and also wanted to make a plug-in for the fact that in Professor Tim Griffin's lab, there is a position available for a postdoctoral researcher. So if anybody is interested in joining the GALAXy team or any of the work that we've been doing, please reach out to him or any of the GALAXy team members. With that, I'll be very happy to take any questions and answer them. Thank you.
Anyone? Anyone? Microphone? Questions? Great.
For transcriptomic and proteomic,
Can you start again at the microphone? A little closer? There you go.
When you are doing your proteomics and transcriptomic data analysis and compare the two, how much variability do you see between replicates?
We have just started doing that study. So the one data that we have for the last drug-resistant cell line, we actually just had 2 replicates. So there is, we are just testing tools right now, looking into that data. But from what we have been told, at least for the technique that's used for proteomic data analysis, I think a variability of 15% CV is what we see. But that's something that we still want to explore using multiple replicates.
Pratik, thank you very much.